Welcome back. This day has truly flown by. I would like to take this opportunity to thank Gerald Samani and William May for their amazing AV support, the NHGRI Communications and Public Liaison Branch for their help promoting our conference and post-production edits. I'd also like to thank Mr. Abhinav Gupta at George Mason for the graphics and webpage support. And a huge thank you to my co-hosts and all of our guest speakers today. It takes a village to make these virtual events successful, and I'm so thankful for our amazing team that we've created. I would like to remind folks that we have our Gather Town space set up, and we will have continued conversation and networking opportunities on that particular platform. When you click the link that's been provided in the chat area, you will be prompted to make a free account to join the space. And then you'll be able to select an avatar and again, use the keys on your keyboard, the arrow keys to navigate the different rooms and look for the signage for whom you would like to speak with. At this point, I'm proud to introduce Dr. Alexander Titus, who will be delivering our closing reflections and future directions to end our Computational Genomics Conference today. Dr. Titus is a strategic business executive at Google Cloud, where he leads healthcare and life science strategies for the global public sector, as well as AI, ML applications for public sector missions. Prior to Google, Titus was the inaugural Assistant Director for Biotechnology within the Office of the Undersecretary for Defense, Research and Engineering, where he led the team developing the DoD's roadmap towards biotechnology modernization. Titus's career has woven between private sector, public sector, and academia, giving him a unique perspective of the landscape for computational genomics. Welcome, Dr. Alexander Titus.
Thanks, Kim. It's wonderful to be here, and thank you, Cami, and everyone else for this awesome conference. It's great. I've worked also with Joe and Stephanie in the past, so it's wonderful to see so many great faces. And of course, to give us all a semblance of normality, I'm going to make the requisite joke about I'm the last person between you and beer in the lobby. So if everyone before you go to the networking walks to your kitchen and grabs whatever beverage of choice, we can make that all happen. Okay. So to wrap up everything, I just have some thoughts about kind of how everything pulls together when we think about computational genomics in the cloud in particular. But I want to start with thinking through how do you get to the point where you're doing computational genomics, and then what do you do after that? So from a clinical standpoint, from a molecular standpoint, from a clinical trial standpoint, how do we get there? So we've had lots of conversations throughout the day about data and the growth of data and how we're leveraging data and the increased funding. But I think one of the things that's just really needs to be driven home is the fact that data, whether it's life sciences data or the social sciences and metadata around our patients, is growing at a compounded annual growth rate of over 60% a year. This is a phenomenal amount of growth. I think NHGRI has on their website that today somewhere between 2 and 40 million gigabytes of data are generated every year in the life sciences, which is just astronomical. And then I also heard another person from the NIH recently say that it's expected that by 2025, the total amount of biological data will be the equivalent to the total of YouTube, Twitter, and the entire field of astronomy combined. So it's just unparalleled how much data that we're dealing with, which really makes us think we need to take a different approach to how we tackle computational genomics. At the same time, one of the questions during the previous panel talked about reproducibility and how do we have quality analysis for reproducibility. And we all know that there's a challenge. 50% of published studies are hard to reproduce, and over 30% of funding goes towards trying to recreate inherently unreproducible studies. And it's because of this, funding agencies are starting to move towards requesting that published works be published in containers and types of technology that make code much more reproducible along with the data themselves. At the same time, we're here having a conversation about cloud because funding agencies are really seeing the value that cloud brings to the researchers and scientists that they fund. The NIH has made large investments in the cloud on behalf of scientists, as well as the NSF and the DoD as well. As Stephanie was just talking about, there's plenty of cloud initiatives ongoing across all of the agencies that are funding research. And this really happens because from an agency standpoint, it helps save money across researchers and actually get better, kind of faster time to results to be able to compute in a way that you couldn't otherwise do previously. All of that comes together with a major trend in the intersection of AI and biology, or computational genomics and machine learning. So not everything is AI. Actually, it's funny, when I was in grad school, I was a statistician. When I moved to industry, I was a data scientist. And then when I moved to Washington, D.C., I was an AI scientist, all with the same background. It's all depends on how we describe the differences. But inherently, there's a major move towards taking data-driven algorithms and a data-driven approach to how we're analyzing biological data. So like I said, just talk briefly about how do we go from clinical data analysis to molecular data analysis to a clinical trial development. So it's not necessarily all in one span of one study, but thinking through the value that cloud computing brings to each of these different components that are really pushing forward all of our fields when it comes to computational sciences. I'm a genomicist by training, so I spent my time working in cancer genomics. I benefited greatly from the Cancer Genome Atlas from the NIH. So all the publicly available data that our funding agencies produce is an amazing resource. So from a clinical data analysis standpoint, let's say we want to take the picture of, we want to identify a cohort of people to then do a subsequent molecular study with. How do we go about identifying that cohort? So you could develop a study ahead of time where you're recruiting patients, but what happens if you have a tissue biobank and you're trying to look for the ability to identify that right cohort across electronic health records, imaging, whether it's radiology or X-ray, and molecular analyses as well. Maybe there have been molecular data that's collected, but not whole genome, and so you're looking for that. How do we get across all of these platforms, all of these data types to find that right cohort? If anyone's done any clinical data analysis along their way to the genomics, you know that doctor's notes are like reading hieroglyphics at times. They're dense, they're abbreviations, irregularities in the writing, and to me, it's basically Latin. So how do you take this in unstructured information and put it together in a way that you can then actually glean the right kind of information as you're doing your clinical analysis and selecting cohorts? So there are plenty of cloud tools that help this happen. And the reason I focus on cloud in this case is that for anyone who's spent half of their academic career trying to reproduce someone else's GitHub repo, one of the beauties of cloud is that you get to use tools that are engineered from a software engineering standpoint. And they're also built with the ability to add on open source technologies as well to enhance that. So in this case, the ability to take a healthcare API and actually query against unstructured data in the same way that you would be using a structured database of information. That helps us, using natural language processing and other forms of machine learning to actually extract entities and information more effectively as we're doing cohort development. But of course, we can't work with clinical data unless we're talking about privacy and HIPAA compliance. And so what about the ability to automatically de-identify information as it's extracted from an EHR system? Similar kind of healthcare APIs are built with the ability to do this inherent de-identification, even in imaging, where it looks for the parts of the image that has identifiable information and either removes or obscures that information to make sure that these workflows are more efficient to be able to go through your data analysis. And then at the end of the day, we run statistics on the study populations that we're that we're using to make sure that we have a representative sample. We don't have bias in our data, but it can be a quick and easier quicker and faster to actually do some quick visualization. So how do we look and make sure that if we're doing a study for the state of New York, that we're making sure we're representing all the counties or all the cities in that state? And so the ability to visualize data quickly helps with that the kind of workflow up front. So I want to give you just one brief case study. The University of Colorado has leveraged cloud technology at a really big scale when it comes to their clinical workflow. So they're using a product called BigQuery as their enterprise data warehouse that joins patient information, diagnosis and treatment information, outcomes, and genetic data, all to create a personalized medical picture of a patient. And so using this information across all these different data types, they're doing analysis across over 6 million patients at a time. And this analysis previously would take up to 8 hours to run this. And now it takes only 15 minutes, given how much scalability that the cloud computing has to offer. So this takes down kind of 90+% faster, and the overall computational cost of doing this is significantly cheaper as well. So if we want to think about from a different clinical standpoint, how about using real-time information in a patient care setting? So in this case, Emory University was developing algorithms to predict when patients develop sepsis 4 to 12 hours prior to a clinical presentation of sepsis, using data that's in real time in the ICU. And so one of the benefits of cloud computing is the ability to do really high quality API integration as well. So not only are you downloading data onto a local system, you can actually interface with clinical data where it's actually collected. If anyone's like me, when I was in academia, I had built a server under my desk. I downloaded all of my TCGA data onto my server, ran my analysis, and I could do that right there. But the ability to do this in real time in an actual clinical care setting is really important. And this helps us then say, what if we want to identify commonalities across patients who are prone to sepsis, whether it's from a microbacterial standpoint, microbiome standpoint, bacterial standpoint, genetic and genomic standpoint, what have you. One other example here when it comes to imaging, there's a good example from Johns Hopkins where the intracerebral hemorrhaging, sudden brain bleed into the brain tissue. 3 million people a year suffer from this, and almost half of them die within the first month of this sudden onset. So how do you develop a workflow that takes advantage of computing in this case to automatically take that imaging and label it? So take and classify what portions of that image are relevant. How do you de-identify using automated processes? How do you convert to all the file types that are going to be useful in your further workflow? Take out faces, make sure your images are segmented, and then put that information back on the image. So at the end of the day, you have a labeled image that shows you the areas of interest for further analysis. And so this can be out of a real-time machine, or it can be out of an EHR repository, but it helps to build this automated workflow. So let's say we have the ability to much more easily do clinical data analysis and select clinical cohorts. Then how do we get into the molecular data analysis side? So I want to focus on one specific tool that has been developed by Google partner company, sister company, Verily Life Sciences, as well as the Broad Institute in collaboration. And it's a secure cloud native tool focused on data, tools, and collaboration. So for everyone interested in data that's existing, publicly available data that's existing to use, Terra currently has almost 2 million individuals with almost 23 million cells represented from a data source, and then has tools built natively with Jupyter notebooks in R in our studio, and in in containers through Docker and things like that. And then over 15,000 people currently using and collaborating on the platform. So all of this is around kind of redefining the paradigm of clinical and molecular data analysis. Traditionally, like I said, I'd download the data from the NIH website, I'd store it on my local server, and all of a sudden there's thousands of copies of the same data all around the world, and you have to make sure it's the right updated data, so on and so forth. But the new paradigm is actually bringing people to a central source of data, and that's one of the benefits that cloud brings to this research world. So Terra is doing just that. Terra really focuses on data production and curation. And so the data generators, biomedical researchers, and tool developers all around a centralized workbench built on cloud services. And the data platform makes it easy to integrate and securely access data from different data types. The collaborative platform makes it really painless to be able to select who is approved and not approved to work with what data. And the tools make it really easy to write your code, whether it's in R or Python or another language, and publish that so other people can use with the data that's here. So Terra right now is powering a number of large collaborations across the NIH and the Broad Institute and then the UK and a number of other areas. Primarily in the community workbench, which is what I was just describing as all the data involved, the NIH All of Us program, and then the Broad Institute single cell portal, where I was saying there's over almost 23 million cells characterized on the platform. And again, the entire idea is to bring data that's collected individually in a lab with publicly available data, with data that comes from maybe an institutional biobank, and then commercial data all into one centralized platform to do analysis. So the NIH All of Us program is a really large program with collaborations across biobanks and genomic partners and healthcare providers and pharmacies, all brought together to do molecular data analysis and clinical data analysis across really large patient populations. So this is starting to get computational genomics at real scale, significant scale. A second application here is the Accelerating Medicines Partnership for Parkinson's Disease. So this is a biomarker discovery program focused on bringing clinical data from 4 existing studies with different protocols and different structures, generating new molecular data across 4 different studies with 3 different sequencing providers, bringing researchers to the data to do analysis, but then feeding those results back into the platform for other people to benefit with for the next cycle of analysis. The primary benefit here is all of these different data types that are supported. And so this is one of the things that cloud provides without having to think of all of the different ways what's the pipeline workflow I built. Was it different from that person? Was it different from that lab? So the ability to collaborate with the same pipelines is really powerful. And so this is really where the AMP-PD program is focused on. If someone's interested in doing a molecular analysis on Parkinson's disease, come to a single source of information where in the hypothesis generating side, there's over 150,000 samples of imputed genotype data with summary stats on over 1 million patients. Once you identify the hypothesis looking to test, then from a molecular analysis standpoint, there's over 10,000 samples worth of multi-omic and whole genome sequencing data, longitudinal clinical data, and that helps to do a hypothesis generating clinical data, then dive into the molecular data, then refine that hypothesis, do additional clinical data. And so this back and forth with all this information centralized makes that workflow much more streamlined and easier to do. So we've done clinical data analysis, done molecular data analysis. What about clinical trial development? So if we're thinking about kind of the ability to quickly recruit patients, make sure we have regulatory adherence to the way we're doing our clinical trials, to make sure that we can then do follow-up studies. There are a lot of really great ways to to collect that information. And so one example is a real world insights program that we've built, developed in consultation with the FDA to make sure it's compliant for analyzing real world clinical data. And the whole goal of this, and this is true for most of cloud tools, is to reduce cost overall and make sure that you're not locked into one type of analysis, because I want to be able to use the tools that are best for the analysis I want to do. From a clinical trial standpoint, automating compliance is really important. And then quickly deploying and running these trials, making sure that you have an automated process to make sure the data is interoperable, and then easily create tools like apps to focus on that. So, for example, the components of this is the interfaces to be able to design studies, the ability to build kind of native cell phone native apps on iOS and Android, and integrate all these tools together. So just two examples real quick. The University of North Carolina has used these kind of tools to develop a mental health and well-being trial to understand how best to support frontline workers during the COVID-19 pandemic. So the protocol design went from what traditionally would take over a year to design and launch to down to under 2 months, with a fully featured app to help patients, or to help public health workers give input on and check their mental health status, and do daily check-ins, which really empowers the end frontline worker to take control of their own mental health. And then last example here is Stanford University with cardiovascular disease, having multi-arm randomized control trials, RCTs, across 100,000 participants, makes it much more simple to bring these, not just to organize data, but to deploy important programs quickly and in a much more streamlined manner. So this is clinical data analysis, molecular data analysis, clinical trial development. They're all part of a spectrum of what we want to do with the computational world. And I know computational genomics is the primary focus of this conference, but I wanted to give some color about how we're integrating in computational genomics alone is one part of a broader set of analyses. And ultimately the reason we do the research we do is to get to the ability to improve patients' lives downstream. So all of this is to say that there's lots of ways to get started using cloud technology. So in Google's case, there's free access for professors and students when you're in class in an academic setting, as well as free online training. There's also you can apply for research credits for up to $5,000 a year for research credits to use cloud technology. And then, like I said, the NIH's STRIDES program and CloudBank programs for NSF-funded researchers help to get involved, answer questions, figure out how to get started. And again, free training with there. And then on bigger analysis and collaborations, there's the ability to have discounted rates and build out those collaborations more fully from a cloud standpoint. So that's all that I have today. I think it's been an awesome conversation. I think that everyone, this is my favorite topic to talk about. I know I'm the last thing between you and your kitchen full of milk, water, or beers, whatever you choose to do. So I thank you everyone for your time.
Thank you so much, Titus, for sharing various health examples. Your presentation has helped us reflect through all sections of the conference, from use cases on what platforms could be utilized and where we could apply for funding to further our research. As you said, we want to make sure that researchers know what tools are available, and that awareness is so important. And I hope through this half-day conference, we have moved one step further in those objectives. I also want to remind everyone that gather.town has opened, so I cannot wait to see you there in person. It's going to be really good. It's a good interface if you have not used gather.town before. So please, you do need to create an account to join the gather.town. The link is available in the chat. So please click on that link, and I look forward to seeing you there. Thank you so much, everyone. Thank you, Titus, again.